Okay, so I want you to picture something. Picture a microscopic architect that has decided to build a sprawling, toxic city inside the most sensitive part of the human body, the brain.
Mm-hmm.
And this architect, it doesn't follow any rules. It just multiplies, invades, and demands resources. We're talking about glioblastoma or GBM.
Yeah, and GBM it's the apex predator of brain tumors. And when you call it a toxic city, that's like a perfect description because it is so aggressive, so invasive.
Right.
By the time we even try to treat it, it has already woven its tendrils into the surrounding healthy brain tissue. That infiltration, what doctors call its malignant migration, that's why traditional treatments they struggle so much.
The statistics are just horrifying. I mean, median survival is still stuck at around 1 or 2 years.
Yeah.
For decades, we have been trying to solve this problem by, cutting, burning, or poisoning these cells, but the results have been heartbreakingly stagnant. But today, we are taking a deep dive into sources that uncover a potential new strategy. We are looking past the genetics of this monster and focusing on something far more fundamental, its deep, dark food secret.
So we're looking for a weakness so profound it's been called a metabolic Achilles heel. Our mission today is to understand how we can actually starve this tumor, not just slow it down.
Exactly.
And the key, it seems, lies in understanding exactly what glioblastoma needs to eat and, critically, how it gets those nutrients in a really restricted environment like the brain.
We have some truly groundbreaking research to unpack. We're talking about detailed cell-level experiments using molecular scissors like CRISPR to knock out specific genes.
Wow.
And powerful new small molecule drug inhibitors like NCT-503 and V9302 that are designed to bypass the toughest barrier in the human body.
It really is an adventure into the unseen world of cancer cell biology. So we're going to expose how this tumor maintains its growth and its shield, all through the obsessive consumption of a single simple amino acid, serine.
So here's the core mission for you, the listener. What if the key to beating the deadliest brain cancer isn't just a bigger bomb, but knowing exactly which utility line to cut? What if we could just pull the plug on its lunch delivery? Okay, let's unpack this and dive deep into GBM's hungry behavior.
Let's do it.
So to really understand the addiction, we have to start with this foundational concept of cancer metabolism. This isn't just a GBM thing. It's like a classic hallmark of every cancer.
Right.
Normal, healthy cells, when they're just resting, they use energy efficiently. They're, running a small electric fan and maybe a single light bulb.
But cancer cells, they completely rewire their internal systems.
Yeah.
They are dedicated to one thing and one thing only: rapid, relentless proliferation.
Yeah.
So they aren't just running a small fan. They're like building a massive factory complex 24/7.
Right.
And that requires an astronomical amount of material.
We're talking about anabolic building blocks.
Yeah.
Bulk materials for new proteins, lipids for all the new cell membranes they have to construct, and nucleotides, the individual pieces of DNA and RNA needed for replication. It's just a continuous, massive construction effort.
And this idea, this goes back almost a century. We can briefly mention Otto Warburg, who observed that cancer cells process glucose differently.
Right, the Warburg effect.
Exactly. Instead of efficiently running it through the mitochondria for maximum energy oxidation, they often shunt it off for quick, inefficient energy generation, which is glycolysis. But more importantly for us, they also divert glucose intermediates into these building pathways.
Okay, now let's focus on GBM's unique threat, specifically in the context of the brain. Normal brain cells are the ultimate sugar users. They rely on glucose almost entirely for just basic function and energy. GBM hijacks this system. It takes that sugar and converts it into specific molecules that actually help the tumor invade surrounding tissue. It's weaponizing the local resources.
And that invasiveness is the central reason GBM is so deadly. When a surgeon tries to remove it, they can only achieve a gross total resection in a percentage of cases.
Why is that?
Because those microscopic, finger-like extensions have already infiltrated the adjacent normal brain parenchyma. You just can't tell healthy brain tissue from malignant brain tissue at the edges.
And as if that wasn't complicated enough, GBM is a tumor of profound metabolic complexity. It's not just one villain. It's a constantly changing gallery of villains.
Right, and this is the concept of intratumor heterogeneity, or ITHE.
Break that down.
Okay, so imagine a complex city where every single neighborhood has a different government, a different electrical grid, and different resource demands.
Okay.
One section of the tumor might be driven by an EGFR mutation, another by PDGFRA, and yet another might have an IDH1 alteration. Traditional targeted drugs often fail because they only kill the cells relying on one specific genetic driver, leaving the other, genetically different populations to flourish and cause a recurrence.
So it's why a single bullet won't work. The tumor just immediately shifts its identity.
Exactly. And the sources point out this leads to other vulnerabilities, like this thing called the cholesterol detour. GBM cells have higher levels of the LDLR protein, that's the low-density lipoprotein receptor. They show a dependency on scavenging cholesterol from circulating lipoproteins rather than making all of it themselves.
That's interesting because that sounds like another form of addiction.
It is, and the sources call this a non-oncogene addiction. And this distinction is really important clinically.
How so?
An oncogene addiction means the cell is addicted to the very gene that drove its cancer, like an EGFR mutation.
Right.
A non-oncogene addiction means the cell is dependent on a completely normal, non-mutated metabolic pathway like cholesterol uptake, just because of the high demand for growth.
So clinically, if we target an oncogene, the cell might mutate to bypass that gene. But if we target a non-oncogene addiction, we're targeting a pathway that, if we shut it down, is just as deadly, but it's not a mutation that drove the cancer in the first place.
Precisely. It's a major vulnerability, and it works for certain GBM subtypes. But the question remains, if they are so diverse, if they have all these different governments and side quests, how can we possibly find one common, critical resource that all the hungry parts of the tumor need, regardless of their genetic fingerprint?
Yeah.
How do we find the one thing they cannot live without?
That, my friend, is the question that leads us to the search for the ultimate building block. Serine.
Ah, okay.
Okay, here's where we pivot from generalized hunger to precision targeting. Forget just glucose, forget cholesterol for a moment. Let's talk about this specific amino acid that has become the singular obsession of the aggressive, relentlessly dividing glioblastoma cell. Serine.
Serine is fascinating because it's labeled a non-essential amino acid.
Right.
And that just means that healthy, differentiated cells can usually make enough of it themselves from other nutrients. But for a cancer cell, a cell trying to multiply exponentially, that label non-essential is a massive misnomer. For them, it's absolutely critical fuel.
Think of it like a massive construction project. You could mill your own lumber on site, but if you want to build 3 skyscrapers in a month, you need a pre-supplied, dedicated stream of high-quality materials coming in 2047.
Okay.
Serine and its partner, glycine, are the ultimate multi-purpose building blocks.
And they are the vital starting materials for 3 crucial, non-negotiable functions. Take one away, and the whole factory shuts down or, explodes from internal stress.
Let's run through the 3 pillars of Serine. The first is nucleotide biosynthesis. Serine provides these vital 1-carbon units.
Oh, okay. What are those?
They're the tiny molecular tags needed to construct the purine and pyrimidine bases, the literal letters of DNA and RNA. Without serine, the cell cannot physically replicate its genetic material. Full stop.
Okay, pillar 1.
Pillar 2 is lipid synthesis. Serine feeds directly into the creation of key cell membrane components, particularly phospholipids like phosphatidylserine. If the cell is trying to double its size and divide every few hours, it needs a continuous supply of membrane material.
Makes sense.
And the third, which is perhaps the most fascinating, is redox homeostasis. This is the cell's defensive shield, its internal body armor against stress.
Right.
Serine is essential for producing the cellular antioxidant known as glutathione, or GSH. GSH is what protects the tumor cell from the massive amount of oxidative stress generated by its own hyperactive metabolism.
So if you target serine, you're hitting the cell's ability to replicate, its ability to build its outer shell and its defensive system all at once.
Yes, it's not just slowing growth, it's stripping the cell bare.
Okay, so how does the cell ensure this massive supply?
It initiates a metabolic adventure.
Mm.
The serine biosynthesis pathway, or SBP. The pathway starts with a common glucose intermediate, 3-phosphoglycerate, which is yanked directly out of the main metabolic highway glycolysis.
This is the exit ramp analogy. So glucose is the main interstate. The SBP is the exclusive high-speed VIP lane that branches off.
And the toll booth operator controlling traffic flow onto this critical lane is an enzyme called PHGDH-phosphoglycerate dehydrogenase. This is the central critical switch.
So PHGDH is the on-ramp operator, just diverting massive amounts of glucose traffic away from just being burned for immediate energy and directly toward anabolic construction making serine.
And here is the key finding that overcomes the ITH problem we discussed earlier. Sources show that PHGDH expression is not just high in some GBM cells, but it is markedly higher in the glioma stem-like cells, or GSCs.
Wait, that's insane. The GSCs, those are the resilient, treatment-resistant ones.
The very same.
They're the ones that survive chemo and radiation and cause recurrence. So you're telling me that regardless of whether the tumor is driven by EGFR or PDGFRA, the ability to maintain stemness, that critical self-renewal and resistance, seems to require this massive, dedicated, serine biosynthesis pathway.
Exactly. This is the universal denominator. By targeting PHGDH, you are preferentially attacking the hardiest, most aggressive, and most treatment-resistant part of the tumor, which just happens to be consistently enriched in this SBP activity.
So when researchers suppressed PHGDH in these overexpressing cancer cells, they saw, what? A strong and immediate decrease in cell proliferation and serine synthesis.
Immediately. It's like turning off the main power generator for the entire factory complex.
No.
And the scale of this diversion is what makes it an addiction. In some of these highly aggressive PHGDH overexpressing cell lines, the serine pathway is so active that it's responsible for nearly 50% of the net conversion of glutamate into a TCA cycle fuel. It fundamentally drives the tumor's entire energy and building matrix.
And we also see how the system is maintained, right? The expression of PHGDH and other enzymes in the pathway like PSAT1 is often regulated by the stress response gene ATF4.
Right.
So ATF4 is like the internal auditor. When the cell senses stress or nutritional limitation, ATF4 rings the alarm and says, we need more building blocks now, and it just ramps up the SBP machinery.
So we have an aggressive tumor that is turbocharging its internal serine production. But this brings us back to your crucial question. If they're making their own serine via PHGDH, why do we still call it an addiction? Isn't the factory self-sufficient?
It seems like they should be. And before we move on, let's just nail down the partner, glycine. Serine and glycine are related, but the sources emphasize that serine is the star player, right?
Absolutely. Serine is the primary driver of proliferation. Glycine, while important, seems to be a secondary fuel. Studies show that while some cells can switch to relying on glycine when serine is completely depleted, high levels of glycine can actually impair proliferation in some cell lines.
Oh.
It's a very tightly controlled system where serine is the high-octane fuel of choice. This highlights the precision needed in targeting this specific metabolic network.
Okay, so back to the addiction question. Why isn't the internal factory enough?
The reason the internal factory PHGDH isn't enough, even when it's turbocharged, is because rapidly dividing cells have an insatiable need. They need so much serine to support their explosive growth that they also have to constantly scavenge it from the local environment.
And this is where the microenvironment of the brain comes into play. The brain is naturally a nutrient desert. Serine and glycine concentrations in the brain environment are significantly lower than in circulating blood plasma.
Exactly.
So GBM cells are basically operating under chronic nutritional stress from the start.
This is the catch. Even the most efficient internal factory needs external raw materials, and the delivery route is controlled by a gatekeeper. Enter the amino acid transporter ASCT2 or SLC1A5.
ASCT2 is famous. It's usually known as the primary transporter for glutamine, another major high-volume cargo ship of cancer fuel. We picture it as the high-volume dock on the cell surface.
The surprise the researchers uncovered is that ASCT2 is also a major contributor to serine uptake, especially when environmental serine is scarce.
Okay, so ACT2 is famous for docking the huge glutamine ships, but in desperate times, when the environment is low on serine, it turns out to be absolutely essential for docking the smaller but critical serine speedboats.
You got it.
So without this dock, the flow slows significantly, compounding the issue created by the low external supply.
The research team decided to perform the ultimate stress test. They used CRISPR-Cas9, those molecular scissors, to genetically knock out the ASCP2 gene, SLC1A5, in aggressive cancer cells, creating ASC2 knockout cells.
Okay.
They wanted to see how the cells would respond under different external conditions.
This is the definitive movie plot moment. So first, the KO cells were placed in normal media, which contained a relatively high concentration of serine, about 285 micromolar.
The result, cell growth was slightly but significantly reduced. It was annoying to the cell.
Annoying.
It told the researchers that the cell had some redundancy, maybe it was using other transporters, or maybe PHGDH was still compensating slightly. The cell was inconvenienced, but not dead.
But then they applied the stress test, which mimics the low nutrient environment of the brain. They placed the KO cells in low serine media, only 50 micromolar.
And the results were night and day. Proliferation was strongly reduced.
Wow.
That data point is the key. It proves the addiction is context-dependent. When external serine is limited, which is the natural state of the brain, the tumor absolutely relies on ASCT2. They even quantified this using mass spectrometry, showing a significant delay in the acute uptake of serine in those KO cells.
So they confirmed the transporter was the bottleneck. And for the ultimate proof, they overexpressed ASCT2, they built more docks.
Right.
And that significantly rescued cell growth, allowing cells to thrive even in extremely low serine concentrations, down to 10 micromolar.
This is why GBM and metastatic brain cells are so uniquely vulnerable. They're already pre-selected to survive this limited environment, meaning they must have highly tuned systems like ASCT2 and PHGDH. Disrupting either one is devastating.
And furthermore, ASCT2 inhibition, even with a drug primarily known for blocking glutamine transport like V9302, causes these secondary catastrophic failures.
Wait, I want to go deep on those secondary failures. We said that serine is needed for glutathione, the cell's defensive shield. What happens when you block ASCT2 with V9302?
Blocking ASCT2 severely limits the import of key amino acids, immediately leading to increased oxidative stress. The research measured this by looking at the GSSG-GSH ratio, the ratio of oxidized glutathione to reduced glutathione.
Okay, let's use an analogy for that. So reduced glutathione, GSH, is the loaded defensive tank ready to fire and neutralize stress.
Right.
And oxidized glutathione, GSSG, is the spent shell casing.
Exactly. When you block ASCT2, the GSSG-GSH ratio elevates. You are seeing more spent casings and fewer loaded tanks. The cell loses its antioxidant shield and is essentially left defenseless against the natural metabolic stress of its own hyperactive replication.
And then it tries to eat itself.
Yes, autophagy, the cell cannibalizing its own contents in a desperate attempt to survive the famine. Researchers saw elevated markers of autophagy in the V9302 exposed cells.
Wow.
And here's the strategic hit. When they combined V9302 with a drug that blocks autophagy, a lysosomal inhibitor, they caused further dramatic decreases in cell viability.
So we starve the tumor, we drain its shield, and then we block its last-ditch survival mechanism. That is a comprehensive, multi-layered attack.
A multi-layered attack, absolutely.
We've identified two critical points of attack: the internal supply chain, PHGDH, and the external scavenging gate, ASCT2. Now let's focus on hitting that internal supply chain with a physical drug, NCT-503.
NCT-503 is a highly specific PHGDH inhibitor, and its promise centers on solving the oldest problem in brain cancer therapy, getting the drug past the blood-brain barrier.
That barrier is essentially the ultimate defensive wall of the brain. It's designed to protect the neural tissue from toxins and fluctuation in the blood. Getting a drug to cross it is incredibly difficult.
It is.
But sources confirm that NCT-503 demonstrated efficient penetration through this barrier.
Not only that, but mass spectrometry analysis showed a substantial accumulation of the drug within the brain, and crucially, the concentration was notably higher in tumor-bearing regions compared to normal brain tissue. This is huge. The drug is attracted to its target and minimizes collateral damage to healthy neurons.
And the target isn't just any tumor cell, it's the GSCs, the glioma stem-like cells, where PHGDH is most highly expressed. This is precision oncology.
And the clinical potential was proven in PDX models, patient-derived xenograft models. This is the gold standard for preclinical testing, and we really need to understand why.
Help us define that for the listener.
Okay, so a PDX model means researchers take a piece of a patient's actual tumor, not just a cell line grown on a petri dish, and implant it into an immunocompromised mouse.
Why is that better?
Because it better preserves the tumor's native architecture, its cellular complexity, and critically, that IT, that heterogeneity we talked about, it keeps the tumor realistic.
And in these realistic models, NCT-503 significantly inhibited GSC tumor growth and conferred survival advantages. But the real game changer was the synergy with radiation therapy.
That combination showed a potent synergistic effect. I mean, think about it. Radiation causes DNA damage and oxidative stress. If the tumor cell's defensive and repair systems are reliant on serine metabolism, specifically for nucleotides and glutathione, and you inhibit that system with NCT-503, you've essentially disarmed the cell right before you hit it with radiation. It can't repair the DNA, and it can't detoxify the stress.
But the metabolic hit doesn't stop at the GSCs. We have to talk about the ripple effect throughout the tumor microenvironment. specifically targeting the blood supply.
Right. The tumor relies on its surrounding support structure, including the endothelial cells, ECs, which form the blood vessels that feed the tumor.
Okay.
Sources show that inhibiting PHGDH in these ECs using another related inhibitor, WQ2301, creates systemic failure.
Tell us what happens to the support structure's defenses.
Remember the three pillars of Serine: nucleotides, lipids, and redox. In the ECs, the redox pillar crashes hardest. The inhibition reduces the total amount of glutathione, and that crucial GSH-GSSG ratio lowers dramatically.
So the defensive shield of the blood supply cells is drained. They're running on spent shell casings.
Exactly. They've lost their internal body armor. Second, the energy currency is destabilized. It attenuates NADH levels, which enhances the NAD plus NADH ratio.
Another super technical term. Let's translate that ratio for everyone.
Think of NAD plus and NADH as the rechargeable batteries of the cell. They are essential for every major metabolic process. When you skew that ratio, you are effectively creating an energy drain. You are inhibiting the ability of the support cells to generate necessary energy, compromising their ability to function and supply the tumor.
So the tumor cell is starved, and the support structure is crippled. This means the therapeutic hit is twofold: direct cancer cell death and collapse of the infrastructure that keeps the tumor alive.
This disruption is so critical that researchers found they could only partially rescue the proliferation of these ECs by adding external antioxidants like GSH or N-acetylcysteine, or by adding nucleosides. It proves that the two major functions of PHGDH in the ECs, maintaining redox and producing nucleotides, are essential for their survival.
This all sounds promising, but this raises a very practical clinical question. We discussed the idea of a double metabolic strike, restricting external serine and glycine diet while using NCT-503 internally. How feasible is that? What are the side effects of putting a patient on a serine-depleted diet?
That's the challenge. The research points to dietary restriction as a potent therapeutic avenue, especially since the brain is already low on these nutrients. Systemic dietary depletion would make the tumor highly sensitive to the inhibitor. But the body needs serine too, especially in the immune system and peripheral tissues. A systemic serine-depleted diet could have significant metabolic consequences that need careful management.
So what's the solution?
You'd need a highly controlled clinical trial, potentially combined with supplements that preferentially support healthy cells without feeding the tumor. It moves us into a complex, personalized nutrition strategy.
We're fighting a surgical war on a cellular menu.
That's one way to put it, yes.
So let's synthesize the grand strategy. We've identified 2 points of attack against the glioblastoma cell's metabolic addiction: the internal supply chain, the PHGDH enzyme, using NCT-503, and the external import gate, the ASTT2 transporter, using something like V9302.
And this transition from broad chemotherapy to precision metabolic targeting is revolutionary. It allows us to specifically hit the one aspect of tumor biology, the serine-glycine pathway, that is consistently enriched in those lethal glioma stem-like cells, effectively overcoming that heterogeneity challenge.
But we have to remain vigilant. The history of cancer therapy is, littered with examples of single-target drugs succeeding wildly, only for the cancer to quickly evolve resistance.
Absolutely.
This high variability in GBM means we have to anticipate adaptation.
And we discussed earlier the challenge of personalized medicine in GBM. Some tumors will be addicted to the LXR cholesterol axis, others to serine, others potentially to something else entirely. The challenge is identifying the dominant addiction in that specific patient's tumor at that specific time.
And when we target serine, GBM won't just give up. It will try to bypass the block.
Absolutely. We have to learn from past therapeutic failures. Think back to the triumph and subsequent challenge of imatinib, which fundamentally changed the prognosis for patients with gastrointestinal stromal tumors, or GIS.
That was the original success story for targeted therapy hitting the KIT receptor.
Right. But within a year or two, resistance was rampant. Researchers found that resistance often came down to secondary mutations within that same KIT receptor. For example, a mutation like V654A might be overcome by simply increasing the dose of imatinib.
Okay.
But a completely different mutation, T670I, structurally altered the drug binding pocket entirely, meaning the drug could no longer dock. That required switching to a second-generation drug like sunitinib.
So, applied to GBM, if we hit PHGDH, the tumor might not just mutate the enzyme, it might dramatically upregulate a different previously minor amino acid transporter to compensate for ASCT2 inhibition.
Hmm.
Or it might switch to a completely different metabolic energy source, like increasing fatty acid oxidation to meet its lipid demands.
That's exactly the prediction the research sources suggest. Future metabolic strategies must be based on combination therapy. We've seen the strong synergy of NCT-503 with radiation. The research also highlighted that PHGDH inhibition was shown to overcome resistance in tumor ECs to CAR T-cell therapy, a groundbreaking form of immunotherapy.
That's a powerful idea. Using a metabolic inhibitor to make the tumor cells weak and vulnerable so that the body's own immune system or the radiation can finally finish the job, we are weakening the fortress before the assault.
It connects the worlds of metabolism and immunology. This level of depth, using CRISPR, metabolomics, and drugs specifically designed to breach the blood-brain barrier, is what moves us beyond just throwing generalized drugs at the problem. We are treating the tumor as a dynamic, complex metabolic machine.
Feels like we are finally thinking like engineers, analyzing the infrastructure of the tumor and cutting off the critical pipes one by one.
That is the final synthesis of our deep dive. Serine glycine metabolism is a deeply embedded dependency in the aggressive stem-like populations of GBM. But because metabolic reliance is highly dependent on the microenvironment, that low serine desert in the brain, we have to constantly monitor the tumor's feeding habits.
So the critical question for the next generation of research, the ultimate challenge, is how to deal with adaptation and heterogeneity in real time.
Right. Can we accurately map the metabolic dependencies of a tumor while it is still in the patient, accounting for its spatial and temporal changes? Because the most dangerous part of the tumor is always the invasive margin, the cells infiltrating the normal tissue. Those cells often have a unique metabolomic signature that helps them survive the harsh environment. If we can identify that unique food signature and starve the tumor at its weakest point before it can switch its food source and guarantee a recurrence, that is the true next frontier of metabolic medicine.
A fascinating challenge, and an adventure that is only just beginning.